Hi everyone, thank you for joining our video on CancerScan Project. CancerScan Project explores smart pathology slide scanners for diagnosis and patient-specific treatment recommendations in oncology. Today, I'm joined by Professor Igor Balaz, CEO of Neovivum and the originator of CancerScan Digital Twin System. As you already might know from our previous conversation, Igor's work brings together computational biology, artificial intelligence, and the modeling of complex biological systems. With CancerScan Project, Igor and his team are exploring how patient-derived organoids and AI-powered tumor digital twin systems could support more personalized treatment decision for pancreatic cancer. Igor, thank you so much for joining our call and allocating your time. Let's start with the concept of CancerScan Project and please tell us a little bit more about unmet medical needs it is aiming to solve, combining biology, artificial intelligence, and the computational technologies to advance precision oncology.
Thanks, Irena, for having me. And I will start with unmet need and then continue with the CancerScan's basic premise and the idea. Our focus is pancreatic cancer, which does not see an improvement in treatments for almost 2 decades. And that's a big difference to other types of cancer. Additional problem is that survival rate is very low and treatment is largely based on trial and error, based on established protocols, but also experience of oncologists that they have. What we want to aim and achieve in this project is to predict response before treatment even starts, and that prediction should be personalized for specific patients. We aim to do it by building a computational model of the tumor, personalized tumor, that predict how it will respond by learning the rules how tumor microenvironment influence, in specific tumor cases, the distribution of drug to tumor and its effect. What is also important to emphasize is that no single technology can solve this. So that's why we are combining organoids, computational modeling, AI, hardware embedding software, and various ontology and semantic techniques in order to bring them together and make something that will be useful in real clinical practice.
CancerScan combines 2 different technologies, patient-derived organoids with AI-powered tumor digital twins. For listeners who may not be familiar, what are organoids and what are digital twins? And why is combining them such a promising approach? So please tell us a little bit more about these aspects.
Yes. Before going to organoids, we need to know that something called tumor microenvironment is one of the main decisive factors in defining how drug treatment will influence pancreatic tumor. Tumor microenvironment is a collective term for the number and ratio and types of different cells that constitute tumor, also combined with their internal communication, vasculature network that connects them. So collectively, they create a heterogeneous environment where drug needs to penetrate tumor, find malignant cells, and kill them. But given all the other things, tumor usually actively defend itself. So we have a very complex problem that needs to be solved and that vary largely between different patients. And that's why we need to recreate those conditions in order to create an appropriate digital model. So organoids are basically lab-grown small 3D versions of patients' tumors. And in CancerScan, we are creating a panel of organoids with different ratios of different constituting cells. We are treating them in a lab with different drug treatments and profile at the molecular and cellular level how the entire tumor responds to those treatments based on different composition of tumors. So yeah, the idea is to recreate as close as possible real physiological scenarios to be able to better understand how those different compositions and communications between cells basically influence efficacy of drugs. When we know those, we can then proceed to creating a digital model that is properly parameterized based on the ground truth. If it is parameterized based on patient's data, then we can call it a twin of that patient's tumor. And I think I at the same time explained why we need to combine them. So if we create a digital model in isolation of physiological ground truth, that's probably an interesting model and nothing more. But we need to connect it with real data, and that's the part of organoid-based characterization that will help us in creating such a model and having basically a right to call it not just a digital model, but a digital twin of specific tumor.
Thank you. So we can conclude that patient-derived organoids are often described as miniature versions of a patient's tumor. So how do they help researchers predict treatment response, and what role do they play in building an accurate digital twin? This is basically continuation of the explanation you already provide.
Yes, so they are essential. Without them, we will end up with a mathematical model with some assumed parameters and some assumed dynamic of what is happening. So it will be just probably interesting model that will not translate well to clinical practice. In this project, we start not with patient-derived organoids because we want to first investigate the scope of interactions between different cells in order to basically create a map of parameter landscape, how we call it, in our digital domain. But they need to provide a proper set of numerical data that will be included into the model to be able to fine-tune mathematical structure to exact physiological ground truth, as I already mentioned.
So artificial intelligence is central to CancerScan, as we understood from the whole discussion we already have had. How does AI integrate data from pathology organoids, and computational models to support clinicians in selecting the most appropriate therapy while maintaining transparency, ethics, and clinical trust at the end of the day.
Yep. I would like first to unravel this question to several sub-questions because it is quite important to make a clear distinction. AI is immensely important, but relying only on AI will probably lead to a black box model that will not be so useful in clinical practice because it will not be able to provide a proper reasoning why some suggestions are made and why some others are dismissed. To remind you and all the listeners, it is of great importance in clinical practice to keep the agency of decision on human beings. So whenever AI suggests something, it needs to be accompanied by a proper reasoning why the suggestion is there. And the oncologist is the final decision maker. He can listen or not listen to those suggestions, but he also or she need a proper reasoning why something is suggested or not. To achieve that, we need to combine AI with so-called mechanistic models, which are basically mathematical models that explicitly model cell dynamic, physiological cell dynamics, their dynamic in response to tumors, and so on. So we have in such type of models, we have a clear delineation what exactly is happening. In contrast, AI aggregate a lot of data, train some weighted response, and give you suggestions without being too explicit about what is happening behind the scenes. So by combining mechanistic models and the AI models, where mechanistic models are providing physiologically relevant boundaries to the AI, we want to achieve that level of openness and explainability of those computational predictions that will be presented to oncologists, so he or she can make informed decisions. And that's I think, one of the most important things here.
Absolutely. So, CancerScan Project also places a strong emphasis on regulatory by design approach and recently has achieved a breakthrough device designation, being accepted into the EMA pilot advice program. So, from perspective of innovator and originator, I would like to hear your opinion on early engagement, on need and importance of early engagement with regulators, and also your perspective on acceleration of development process and the journey from lab to clinic.
Yeah, thanks. If you say acceleration, I would add that it can sometimes mean complete negation of the progress if regulatory by design is not started and included properly. Why? We first need to have clear idea what our aim is. If we want to achieve a nice academic program that will develop a brilliant model that do something, that's fine. but the regulatory environment reality is something different, and the level of data for validation is a different beast compared to purely academic research. So if you do not take those moments from the outset into your consideration, you can end up in a corner without ability to progress really towards clinic, even if you have a proper model, because you will probably need to collect all the data again or to redesign parts of the model. So a lot of problems are just waiting to emerge if you do not plan it properly. In our case, if we are building a digital twin that will be used in clinical practice, we are talking about something called software as a medical device. And from the outset of the project, we need to know what exactly are regulatory demands for a medical device. And we need to consider our model not just as a model, but as a medical device that will be used in a practice. So to summarize, if we do not take those into account early on, we can end in some nice but clinically useless results. And I think that's the main lesson, because it will take years and a ton of money to produce nice papers and then nothing more.
Thank you so much, Igor, for this comprehensive response. And I'm especially grateful, being a regulatory science expert, to hear this approach and this approach in innovations from innovator like you. Thank you so much.